name is Rico I'm a third-year PhD student in the Forney Lab at Albany, and today I'm going to try to remind you of this elusive structure that we all have known as the terminal nerve. So first I'm going to go through the basics of puberty. So puberty is a postnatal developmental process that's going to cause various physical and psychological changes that we must undergo to reach sexual maturation and propagate as a species. So these signals actually come from the brain and signal to the gonads, and they originate or come from a group of neurons known as the gonadotropin-releasing hormone neurons, or GnRH neurons. So these GnRH neurons work to establish this hypothalamic pituitary gonadal axis, or HPG axis, and this is important for both puberty and fertility. So here's a picture of the pathway here. The GnRH1 neurons are in the hypothalamus of the brain. They signal to the gonadotrope cells within the pituitary gland, causing the release of follicle-stimulating hormone and luteinizing hormone. These then signal to the gonads to release the sex hormones and then stimulate downstream pubertal effects. Now I'm going to talk about a condition related to puberty in humans. So one condition is known as hypogonadotropic hypogonadism, or HH, which is going to lead to a reduced release of the gonadotropins, follicle-stimulating hormone, and luteinizing hormone, which then downstream leads to a reduction in the sex hormones, leading to delayed or absent puberty. And this whole condition is actually caused by defects in the function of the GnRH1 neurons. So this condition actually has two versions or variations, one known as normosmic idiopathic, HH, which has a normal sense of smell and problems with puberty, and Kallmann syndrome, which has problems with both puberty and sense of smell. And these problems with sense of smell also come in different degrees as well. One known as hyposmia, which is a reduced sense of smell, and anosmia, which is no sense of smell at all. So what is the link between puberty and sense of smell? Now I'm going to talk about how discovery in developmental biology led to this linkage. So here I'm showing a parasagittal or a side or a profile view of a mouse's head, and just to orient you around the olfactory structures here, we have the vomer nasal organ in the nose, or the VNO. Connected to that is the olfactory epithelium, and then towards the brain we have the olfactory bulb. So what was discovered is that the GnRH1 neurons are actually originate or are born within the nose. They propagate here, and as development progresses, they need to make this migration from the nose to the brain and hypothalamus to achieve their function. So that's where this linkage comes. The GnRH1 neurons are very important for puberty, and they need to make this migration from the nose to the brain. So our lab asked the question if the development of the olfactory system and the development of the GnRH1 neurons directly linked. So in this first cartoon, I'm just showing normal olfactory development, where the GnRH1 neurons successfully migrate from the nose into the brain. And on the right here, I'm showing an immunofluorescent staining around the olfactory bulb in the brain. And in green here are the GnRH1 neurons successfully invading the brain along peripherin positive axons in red. So in this study, we looked at two different mouse models that have aberrant olfactory development. They actually lack an olfactory bulb altogether. So we want to see if the GnRH1 neurons can still make it to the brain. And from this staining, we can see that despite no olfactory bulb, these GnRH1 neurons can still make it to the brain, as you can see all of these green neurons here. So if the olfactory system is not needed, is there a different nerve that these neurons need to be guided by? So that is the basis or hypothesis for my project, that these GnRH1 neurons, which again need to migrate from the nose to the brain, migrate along or are guided by the axons of the terminal nerve. So what is the terminal nerve? The terminal nerve is a cranial nerve that was discovered about 100 years ago from dissection experiments from various organisms such as humans, fish, and rodents. So the only thing we know about the terminal nerve is that it projects from the nose to the basal foregut. It's the only cranial nerve that does this. We don't know its function, we don't know what genes it expresses, and we don't know what it looks like during development. So now I'm going to show you some of my lineage tracing experiments of the terminal nerve and show it for the first time during development. This data is also unpublished and confidential. So I was told to say that. To start off our design experiments, we looked at genes linked to Kallmann syndrome, because we believe that the terminal nerve is what's being affected in Kallmann syndrome patients, leading to those GnRH1 neuron defects. So we looked at two genes of interest, proconnecticin 2 and proconnecticin receptor 2, or ProKR2, And what's interesting about these is that ProCare2 is one of the most downregulated genes in Kallman syndrome patients. And in ProCare2 knockout mice, they lack an olfactory bulb and have GnRH1 neuron defects similar to Kallman syndrome patients. So I'm showing in situ hybridization of ProCare2 mRNA expression. In purple here, we see cells in the BNO, this mass or blob leaving or outside of the BNO, and then we have near the forebrain junction, these cells also stained. They appear to be invading the brain. So who are these ProCare2 cells? We designed experiments with this question in mind using the mouse model ProCare2-Pre. This is basically going to trace any neuron that expresses ProCare2, and we will be able to see where it goes. So can we trace the terminal nerve using this model? So for our first experiment, we looked at an early stage E11.5 or embryonic day 11.5, and in green here, I'm showing HUCD, which is a neuronal marker. What you can see here is that many neurons are being born and currently migrating. And in magenta is the tracing. So what's very interesting here is the only tracing at this early stage we see is near where the olfactory bulb would be. It's not formed yet. So this suggests that these may possibly be pioneer neurons. So pioneer neurons are the proposed first neurons to form in the olfactory epithelium, touch the brain, and induce olfactory bulb morphogenesis. So we confirmed that these trace cells in magenta are in fact neurons via HCD positive or HCD staining. As you can see, they co-localize, forming this white here. So this is an interesting observation about the pioneer neurons, but I have not actually shown you if the terminal nerve can be traced using ProCare2. So is this possible? And the answer is yes. So 2 days later at E13.5, we see this tracing has increased exponentially. We see tracing within the BNO here, tracing in the structure known as the Grunberg ganglion, which is a chemosensory structure important for pheromone detection. And then coming out of here, we see a stream of tracing going towards the middle of the nose, into the brain, and then branching in the basal forebrain. So we believe this to be the first image of the developmental trajectory of the terminal nerve. And when I line it up with the cartoon of the model we had earlier, it lines up nicely. So our next question was how the GnRH1 neurons interact with these trace neurons. And what you can see here in green, again, the GnRH1 neurons migrating out of the BNO almost along these trace fibers of traced ProCare2 fibers. And in the higher mag image I have here, what you see is these GnRH1 neurons are migrating almost right on top of these ProCare2 trace neurons, suggesting them to be two different populations. The GnRH1 neurons themselves are not traced, suggesting them to be two different populations of neurons that are migrating out from the nose into the brain. So the next question we had was, if these trace neurons are the bulboneurial sensory neurons, another population of neurons within the nose. So here I'm showing it at E13.5, the BNO, this like oval shaped structure here, and in green and cyan are the bulboneurial sensory neuron markers, MACE2 and H2 epsilon. So what you can see here is that in magenta, the trace neurons actually do not co-localize with this staining at all, suggesting them to be a different population of cells. They're actually missing spots where the trace neurons would be in this channel here. And then we did a time course, so over 3 days we see the same or similar result. And from my quantifications, I actually have found that an average of 90% of the trace neurons across these 3 days are negative for the PSN marker expression, suggesting them to be a distinct population of neurons. Now, finally, I'm going to talk about my single cell sequencing experiments of embryos in an attempt to get cells of the terminal nerve. So in our first approach, we looked at wild type E14.5 noses. We dissected the noses out, associated cells, and then sequenced them in an attempt to get cells of the terminal nerve. So what we got are various populations of cells within the nose, including the respiratory epithelium. This cluster we've termed the primordial BNO. It's very early cells of the BNO, neuronal progenitors, olfactory sensory neurons, the Grunberg ganglion, and finally one of the first clusters of the terminal nerve cells here. But it's still somewhat small. So can we do better? Can we isolate more terminal nerve cells? So we took a second approach where we used our ProCare2 trace embryos, again dissected the nose out, associated cells. Now we have two different types of cells. We have fluorescent and non-fluorescent cells, and then sorted them via fluorescent activated cell sorting or FACS. So we only get the fluorescent cells. So from those experiments that I showed earlier, basically we are taking only the trace cells, we should only be getting the terminal nerve. We took our unsorted or wild type data that we had earlier and combined it with this sorted data set. So now in the magenta, I'm showing the sorted data set. We got this large cluster belonging to the Grunberg ganglion, which itself is very interesting because it's close to the terminal nerve cluster, and this cluster here of the terminal nerve cells. So now we have two different versions of the terminal nerve cluster. We have the non-sorted or the wild type and the sorted cells of the terminal nerve. So we looked at the enriched genes, for some enriched genes of this cluster, and we found 3 of interest. All 3 of these have potential roles in fertility, which could give insight into the possible function of the terminal nerve. And all 3 of these have similar mRNA expression patterns via insect internalization. What you see are clusters of cells outside of the VNO that appear to be going towards the brain. So this is interesting in that they could be potential markers for the terminal nerve in the future. So to summarize, what I've told you today is that for the first time we've been able to trace the development of the terminal nerve using ProKR2. And there are two distinct populations of neurons that migrate from the nose to the brain, the GRH1 neurons and the terminal nerve neurons. They migrate very closely together despite being 2 different types of cells. Within the VNO, we have the ProKR2 trace neurons and the vulvar sensory neurons, 2 distinct populations of neurons again. And for the first time, we were able to isolate the transcriptome of the terminal nerve, and terminal nerve cells were sequenced for the first time again via 2 methods, both unsorted and sorted. So before I stop, I'd just like to give a shout out to my friends in the lab, Nikki, Noah, and Ed. They'll have posters here about different aspects of olfactory development, and they've been working hard to show you some really cool stuff. So with that, I'd like to thank my lab, especially Ed and Charisse, for the help with this project, and I'll take any questions. It could be, so what we're saying, the ProKR2 trace neurons are the terminal nerve, so they could be migrating together because they express the necessary guidance used to stick together, or there could be another structure like you're saying. But we speculate that these ProKR2 trace neurons are the terminal nerve.
Is there a student question that I should not preempt? Just a technical note, with your fact-sorted neurons and your just isolated global picture, I'd be a little concerned that the fact sorting would change the transcriptome and we might have some artifacts creeping in that create different clusters. Do you have some kind of control to look at to know if those clusters really are different or might be an artifact of the facts?
Our control would be that they do express similar genes. They're 2 different versions of the terminal nerve cluster, but they still cluster together when we combine them. So, and they do have similar enriched genes expressed when I compare the 2.
I might have missed this, does the terminal nerve project first and then the GNRH neurons, or are they going at the same time? Sort of what's the timing of this?
So that's the interesting thing. We actually, we believe that the terminal nerve projects first, and then the GRH1 neurons have to use that to migrate along and get into the nose of the brain. But we don't actually know this. All right, thank you.